And the next speaker is Frank Elbert from University of Minnesota, and back to talking about the 1,000 yeast lines, I believe.
All right, thanks, Mike. Seems yeast only come in high numbers today. Good. So thanks a lot for this. I'm going to be talking about a collaboration between my own brand new lab at the University of Minnesota and my postdoctoral lab with Leonid Krugliak, where I've been working shoulder to shoulder with, the mouse doesn't work, well, with Josh Bloom on the left here, who is a research scientist with Leonid. And we'll be talking about the genetics of gene expression variation. So just very briefly, by way of introduction, you're all aware that in any species where one has looked, genomes are chock full with regulatory variations, so DNA differences that influence gene expression levels between individuals. And there's a lot of excitement around those regulatory differences because they're an important source of variation in complex quantitative organismal traits, such as the risk for common disease, but also fitness and things like that. So regulatory variation can then be mapped as expression quantitative trait loci, or eQTL, and that has been done in many species by now. But we know that in many species that we're missing many eQTL. So we found many, but we're also missing many. And we can know that based on genome-wide methods of estimating the heritability of gene expression. And this is just an example from humans, where if we look across genes, typically gene expression heritabilities are in the range of 20%, so that's how much genetics there is. We can break that genetic component down into a local variation, which sits close to the given gene of interest, and we typically find that around 30% of that of the genetic variation comes from this local variation, and the rest comes from other positions sitting elsewhere in the genome acting in trans. Then we can ask, so this is sort of how much genetics there can be, so how much of that have we identified with individual eQTL, and in humans at least, we've done a pretty good job for the local component there, but for the trans component, we basically have very, very little idea. It's a little bit better in model organisms, but generally, we don't know too much about that yet. So this is where this project came in. We then wanted to do a very highly powerful, genetically and statistically powerful experiment where we try to understand almost completely where the genetic variation for gene expression levels comes from. So we took two strains of yeast, a lab strain and a wine strain that differ from each other at about 1 SNP every 200 bases or so, crossed them, made a hybrid, let that hybrid undergo meiosis, such that then each of the recombinant progeny that comes out is a random combination of alleles from the two parents. We took 1,000 of these individuals then, and then using a robot-assisted pipeline did RNA sequencing in all of those to map expression QTL. Why 1,000? Well, we knew that statistical power would be high in this design. You see power here on the y-axis as a function of effect size. Our design is the red line, and we can see that we're basically perfectly powered down to 2% variance explained. And by way of comparison, the blue line there is an earlier experiment that has been done in the same cross using about 100 individuals. So you can see our power is a lot higher with this data set now. So what did we see with that? Well, across the 6,200 genes or so that are annotated in yeast, we found almost 34,000 eQTL at a reasonable false discovery rate. If you put those together, that of course must mean that a typical gene is influenced by several eQTL. We're finding a median of 5. Whereas in this earlier, smaller data sets with 100 individuals, we found 1 eQTL per gene on average, right? So that's a pretty big difference when you think about it, because before it seemed a typical gene is influenced by 1 eQTL somewhere, but now we're finding several positions all over the genome that talk to any one particular gene. Importantly for us, these eQTL that we found are sufficient to explain the majority of the genetic variation that there is in this cross. You see that here, every dot, black dot is a gene, and on the x-axis is this genome-wide estimate of heritability. This is how much genetics we can hope to explain. This is how much, that's the maximum there is. And on the y-axis, you're finding the variance explained by those average 5 eQTL we're finding per a typical gene. So you can see that the eQTL track the genome-wide estimate really well, and on average we're able to explain 78% of this heritability, which means that the eQTL that you're about to see are in some sense complete, right? So we're approaching an exhaustive catalog of where the important gene expression variation in this cross in yeast sits. So this is then where the eQTL are. For those of you who are not familiar with reading a plot like that, On the y-axis, we have physical gene position. On the x-axis, we have the position of the eQTL that influence a given gene. When we have a local eQTL, for example, variation in the promoter, then x is the same as y, and we get a dot on the diagonal, and then the diagonal is all the local variation for all the genes. And all the transacting things where the eQTL is elsewhere in the genome are then off the diagonal. And we're going to be spending the rest of this talk circling through the various components of this map, beginning with the local variation there. The main thing to say is that there's a lot of it. 30% of the genes have an eQTL at genome-wide significance. When we drop that threshold down to a point-wise significance test, so one test per gene, we find that 70% of genes are linked, have the expression linked to their own position. Keep in mind, this is only looking at 2 strains, right? We're not looking at the population samples as in humans. This is only variation that comes from 2 different parents. We can look at effect sizes of the local versus the distant eQTL, and much like what Trisha found for her mutations, we see that the local eQTL do have larger effects. You can see on the left there in red and right shifted. But because there's then several distant eQTL per gene, they do add up to explaining more variance in total in gene expression, again illustrating the importance of this transacting variation there. So let's then talk about the transacting variation a little. If we focus on the off-diagonal things, then of course the most striking feature here is that we see that there's a very highly non-random distribution, right? So there's a lot of positions where a single locus in the genome influences the expression of many, many genes. Most of the distant eQTL fall into one of these hotspots. 90% of them overlap a hotspot. And this is at this point, it's worth taking a little bit step and reminding ourselves that something that didn't happen here, right, is that this map did not fill in completely. So there was a chance, because there's so many genes and so much variation, that each of these variants might have been a trans eQTL for something some of the time in a detectable fashion, but it didn't happen. So with this almost complete accounting of where the distant variation is, this trans architecture is still very much dominated by hotspots. So another way of looking at the hotspots, just asking how many genes they affect. This is how many genes link to a given bin in the genome. And the way to take what to take away from this slide is that some of these hotspots really have extraordinarily wide-reaching effects. Right? You can see that some of them affect thousands of genes. Keep in mind, yeast only has 6,000 genes or so. So some of these affect half the genes in the genome and have very, very wide-reaching consequences. Many of these largest hotspots we had seen in earlier work with in the smaller panels before. These are the gene names in cases where the gene has been validated by allele swaps and other experiments. But of course, what's new here is that we now see how wide these hotspots really reach into the cell. We're then, of course, mostly interested, or also really interested in all these new hotspots that we found. So we've come up with a little algorithm to combine information from all the gene expression traits that link to a given hotspot and try to use that information to fine map as well as we can the position, to get at that position that jointly explains expression among these genes together. And just by way of illustrating that, for this one new hotspot here on chromosome 8, it's a little hard to see, so we can zoom in on the chromosome, and we're trying to fine map this guy on your right over there, so this thin guy there. When we then apply our algorithm to that, the confidence interval for the position happens to land in a single gene as a transcription factor, SDB5. And then we can make sure we're actually right about this guy. So we can look at the genes that are influenced by this hotspot and ask if their promoters are enriched for containing transcription factor binding sites for this transcription factor, and that's what you see here. H dot is a test for one transcription factor, and you can see that SDB5 is really highly enriched up there. So we're pretty sure that in this case we picked out the right gene for this new hotspot. Now, admittedly, we don't always do so great across all the hotspots. This is the number of genes that we're finding per hotspots. We can talk later in the questions, if you like, about why I think some of these hotspots on the right there do still have very large confidence intervals. But with that said, there's still 18 hotspots that we're able to map to less to 5 genes or fewer. We can then begin to take those 54 genes and ask, is there anything in common about them? Ask this question, what is the nature of genes that tend to underlie hotspots in this cross? And when we do that analysis, the top term for molecular function gene ontology that pops out is transcription factor activity. That, of course, is great and makes a lot of sense. The thought is that we have a mutation in transcription factor, and that leads to a change in the targets of the transcription factor. What's interesting about this is that in these earlier, smaller data sets, when the hotspots that we had were fewer and they were wider, this enrichment was actually not seen. Whereas now with this better powered view, we actually see them. Other things we see come up are things such as cell communication, response to nutrients. Here then the thought is that the causal mutation sits maybe in a sensor for a certain nutrient or in a metabolic enzyme, and then that either perceived or real change to the cell leads to a transcriptional cascade that shows up as many genes linked into those positions. There's just actually one final point I would like to make, and this is just a final time looking at our 2D map here, and I want to draw your attention to, and I really can't get this mouse to work. Well, I want to draw your attention to these. You see these regions where there's really not any trans eQTL, right? That's a corollary of you have hotspots. They take most of the trans eQTL, but there's positions where there's just nothing linking in trans. So there's trans deserts, if you like, right? But if you look at the diagonal, there's no local desert. So there's no stretch in the genome where there isn't local variation. So think about what that means, right? So here we have genetic variation that's changing the expression level of genes locally in a manner that we can detect, but we're not finding other genes responding to those expression changes, at least down to a 2 or 1% effect size as we can detect here. So this is really in some sense saying that at least in ways that are detectable at the transcriptional level, it seems in a way the cell doesn't seem to mind all that much that genetic variation has bumped around the expression levels of these genes to some extent. This, of course, opens up a whole set of questions that Tricia also started talking about. What are the consequences of these small expression changes, such as those that are actually caused by genetic variation? Of course, has raises other questions. For example, in human eQTL studies, where there is a large focus on these local eQTL and overlap with GWAS studies, then is of course a big question. But here we're seeing that many of those might just not have consequences, and then the question becomes which of them do. So with that, I'm going to close. I'm going to just put up this short list of bullet points with a blunt disclaimer that I'm still looking for postdoc. The lab is growing. Some people have signed up, so now would be an excellent time to join the lab as it's getting started. Thank you very much for your attention.
In cases when you have multiple eQTLs affecting the expression of a gene, have you looked at in what % of cases they are actually annotated in the same pathway in something like KEGG or reactome? I mean, to what extent do we know the mechanism of the interaction of these eQTLs?
Right, so you're asking for the case where several eQTL influence the same gene?
Yes.
So in order to do that well, we would have to know the causal genes in the eQTL, and we have great to vague guesses depending on which of those eQTL are. So I want to say that's a good question. I don't have a fantastic answer for you yet.
Was the growth condition YPD?
It was YNB.
Okay. Have you looked at the sort of the diverse set of physiological states? Obviously, you can't do that for all your 1,000 and sequence them, but there's a vast amount of transcriptional data, and you can compare that to some of your deserts, and can you look for, in some of these deserts, are those genes most needed in specific biological conditions, and that might explain why you're not seeing them.
Good idea, have not done that yet, yes. Multiple conditions is a great idea. This is perhaps not the way you would want to go about that.
Hey, you mentioned the trans-eQTL hotspots, but there are also trans-eQTL which maybe affect one gene or something. Do you know anything about what those are?
Can you repeat the question?
So you talked about trans-eQTL hotspots. I assume there's other locations where you have a trans-eQTL, but it only affects like one gene rather than affecting 50 genes. Did you look at those at all?
Yeah, we still want to look at those in more detail. So far we've been focusing on the hotspots, and I mentioned that 90% of the trans-eQTL do overlap where the hotspots are. It's a great point, right? Because the question is, are there sort of really super specific effects, right? Where you have something that only affects one gene or maybe two genes in a very localized, functionally localized fashion. And we want to look for those. We haven't done that yet, to be honest. We've been focusing on the hotspots so far. Of course, we will put all this data out there when we write the paper and hopefully preprint, so then you're welcome to play.